So today is 4/25. So we're working with the codon chart just so that you have a refresher on how to use it. Remember the 4 bases of DNA. Cars park in the garage and apples grow on trees. So it's C to G, A to T. Now, if you have a section of DNA, like our little section of DNA here, and you want to make a protein from it, what has to happen? Unzips, and each of those sides gets a complement made of RNA. So I'm going to use a different color for RNA, and let me put in some other letters here. T, T, C, G, A, T. Okay, so there's my section of DNA. I'm going to vaporize this other half. When the RNA comes along to make a complementary strand, some of those bases are the same, but there's one that's different. So C pairs to G, same as in DNA. Oops, and we hit the different one right away. There. Is that easier to read? What would be the easiest color to read? Black? Yes. So C pairs to G. Remember what A pairs to? U. Because there's no T in mRNA. So A pairs to U. Apples grow on trees. You like apples. Get it? Apples grow on trees. You like apples. So when we're doing, so this is our DNA. This is our RNA. What pairs to T? A. Okay, so A. What pairs to C? G. What pairs to G? C, U, U, A. That's our strand of mRNA. And that mRNA is what determines the amino acids. Remember that the tRNA, the anticodon, is just like the license plate on the truck. It doesn't matter. It just sort of confirms that the delivery information matches. So you're pairing based on the mRNA. So if we just practice using this first 3-piece chunk, what do we call the 3-letter chunks of mRNA? Codons. So if we practice using that first 3-letter codon, we're starting with G, because that's our first letter. So on a circular DNA chart, you start from the middle and go out. Our next letter is U, so we're going to go here. And our next letter is A, which gives us valine. And as it turns out, it doesn't matter what the last letter is. Those all code for valine. Okay. If we look at the next 3-letter chunk, AGC, we start here, A, go to G, and then there's our C. That would give us serine. Okay? So on the chart that you have, you're being asked to fill in either the DNA triplicate from the mRNA, or the tRNA anticodon, or the amino acid. Okay? One thing that they didn't mention that you'll need to make a note of is that methionine is your start codon. Okay? So I would write in there that methionine is the start codon. So I'm going to give you, actually they had about 30 minutes yesterday to work on this because the network wasn't working, so we couldn't go over test stuff. So I'm going to give you time in here to work on this worksheet and this study guide. These are going to be due tomorrow. They're both some points. Some of you need some homework points. I am still diving through the projects. I'm putting them in as I go for the most part. Nothing more I can say about that. So, okay, work. I'll put on the radio. Okay, so let's actually do the first one together, since apparently I had a brain spasm right in the middle of that and didn't finish what I said I was going to do. I like this worksheet because it's asking you to go back and forth from DNA to mRNA to tRNA and all the way back. And some of the air test questions I've seen ask you to do that same thing. They ask you to think about, from the DNA, what's the mRNA? How could you change the mRNA by changing the DNA? So that back and forth logic is a good thing. So for this first one, we have AAG is our mRNA. what DNA would have coded for that? T, T, C. Well, what would be, and again, I kind of like the metaphor of the license plate on the truck. What would be the tRNA anticodon that would mark the tRNA that's carrying the amino acid over to the ribosome? Well, it's the complement to AAG in RNA bases. U, U, C. The tRNA anticodons are going to look a lot like the DNA triplets. The difference is going to be if there's any thiamine in the DNA, you're going to see U's instead of that. So, and then you got to go through the process of looking up the amino acid. So A, G gets you lysine. You look it up from the mRNA, yeah. And that's a really common misconception and an important thing. The mRNA is what codes for the amino acid, not the tRNA. The tRNA is just the license plate on the truck. That's just like checking to make sure you got the right thing when it gets delivered. Okay? DNA here. We've got the matching base pairs. We've got each side that's sugar backbone. Where is this stored? In each cell in your body, in the body of a maple tree or a daffodil or a banana or a spider or a chameleon, where is this stored? In the nucleus. Yeah, it's stored in the nucleus, and it's organized, it's wrapped around what? To keep it sort of neat. Remember? It's wrapped around proteins. So it's wrapped around proteins so that it's in sort of this neat form. When the cell isn't dividing, it's longer and skinnier. As the cell gets ready to divide, it shortens up into those chromosomes that we've been looking at and talking about. Okay. When it splits, half of this whole thing kind of goes away. It unzips is what it does, as a matter of fact. And it only unzips part of the DNA, so it really only unzips the piece that's needed to synthesize a protein at one time. That mRNA copy then that's made, I'll make my little mRNA copy here, so we have U, C, G, A, G, G, C, 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 U, A. That mRNA copy heads out into the cell, out into the cytosol. Where does it go? What's it aiming for? It gets fed through a particular little organelle in the cell, which is called a ribosome. So it gets fed through the ribosome, and then I'm going to erase my DNA altogether here. Little sort of delivery vans that are carrying sort of, again, I keep saying license plate, but it's a good metaphor. So that would, if I'm looking at this one, that would be A, U, U. Those little pieces of tRNA come along carrying amino acids. And based on the codon that's being read, they start hooking up, basically dropping off those amino acids, and then they go back and get more amino acids and sort of circle around the ribosome until they're needed for delivery. And this whole piece of mRNA will get fed through the ribosome. They talk about ribosomal subunits. There's, you know, we could say the big half and a little half. They call it the large subunit and the small subunit. This is how your cell makes what? What chemical are we making here? Proteins. And these amino acids end up bound together with peptide bonds, so they're sometimes called polypeptides. Okay? Okay. Why does your cell need to make proteins? Just 'cause? What do these proteins do in your body? Or in the body of a maple tree, or a daffodil, or a mouse, or a chameleon, or a tarantula? Huh? Well, what do they do? They do a bunch of different things. So If you have the ability to clot your blood, that's because of proteins in your blood that are sticky. And that when there is an opening, your body senses that, it sends those proteins to the site, they stick together, they form a clot. If you are able to move fluid out of your lungs, then that's because of a protein in the surface of the cells of all your lungs that allows your lungs to move mucus. So there are a lot of things that proteins do in your body and build in your body. Your skin is composed of a lot of kinds of protein. Your muscle is composed of protein mass. Okay, we're going to do a quick recap video on protein synthesis. This is 4 minutes. Okay, so as we think then about cells building proteins, running protein synthesis, the question that I have for you, so tomorrow when you walk in, the study guide is due, the Codon worksheet is due. Okay. You don't have to do dosary, you're just going to turn them in on paper. What we're going to start talking about tomorrow is cellular functions. So they shifted the standards a little bit when they updated the standards. And, you know, we used to spend a lot of time having students memorize parts of the cell and what they did, blah, 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 blah. And they kind of shifted the standards to say it's more important, and I like this, it's more important to think about what the cell does, how it functions, and how it interacts with other cells, with other parts of an organism, or with the environment, than it is to just memorize the parts of it. So we're going to talk a little bit about the parts of the cell, but we're going to talk about those in the context of what the cell needs them for. So protein synthesis is a good place to start because a lot of cells do manufacture, well, all cells manufacture some kind of protein. So the thing that I want you to think about, that looks like some sort of horrible face, doesn't it, is what does a cell have to do? What is required of a cell? Any cell. Bacterial cell, your cell, cell in a maple tree, cell in a blue whale, cell in a mouse, cell in an earthworm, cell in a daffodil, cell in that fern. What does a cell have to do? Okay, make protein. What else? What do you have to do every day to survive? A cell's it's a tiny, tiny, tiny living thing. Now, your cells can't live on their own. Some single cells can, but they have a number of things in common, basic common commonalities of all living things. What does a cell have to do every single day to survive? Okay, let's say control water balance. So that's part of homeostasis, maintaining your water balance. Too much water and the cell pops open. Not enough water, it shrivels up and you got cell jerky. Homeostasis. Okay, maintain water balance. What else do they have to do? Somebody else.
Store the DNA.
Okay, they got to store DNA. But that's kind of like, you know, stick it in your locker and forget about it. It's not like an active thing like every day. Okay, I got to store my DNA. You know. So what are things that they have to accomplish on a daily basis that you can think of? Any of you going to lunch soon? Why? Why do you go to lunch, other than the social aspects? Huh?
I don't even know if I just
Okay. Some of you don't go for the social aspects. You're all about taking in energy. What do cells have to do every day? Take in energy. I mean, it's not eating like we think of it, but they have to somehow obtain energy. What else do cells have to do every single day? Anybody here got a dog at home? Only a few people. Okay. Does your dog stay in the house all day long, never goes out? Can your dog stay in the house for a week at a time? What would happen? What would happen? Go ahead, say it. They'd poop on the carpet. They'd poop on the carpet. Or worse. I don't know what's worse, but yeah. Dogs eliminate waste. You eliminate waste. Guess what cells have to do every single day? Eliminate waste. Or they drown in their own waste. Is there anything else you can think of that a cell might have to do? I'm not saying that this is a complete or incomplete list. Are there things that we have not said that you think, oh, well, a cell would probably have to fill in the blank? Rest? Oh, that's a good question. Okay. We'll put a question mark there. I know the answer, but we'll explore that a little bit more.
Well, I wonder, is how do cells and things like know where they're going to factor they are?
Like single-celled organisms? Very often they don't. So single-celled organisms in some cases have what we would think of as primitive photosensors, so they can kind of move towards or away from light. But in a lot of cases, single-celled organisms don't have any sort of sense of control over where they're going. They're just moving. Reproduce. That's a good one. Only if we want to have more cells, we do. Anything else? So take in energy, get rid of waste, reproduce, rest, store DNA, maintain water balance, make protein. Is protein the only chemical used in your body? No. What other chemicals do you find in your body? Water. Yeah. Others. Hormones? Neurotransmitters? Who here has ever felt joy? Oh, please raise all your hands, because that would make me too sad if you had never, ever experienced joy in your whole life. Okay. I'm just making sure. Okay. Donovan, did you raise your hand? I'll assign you homework if I have to. So, joy. Guess what it is? Okay, you know how I say I ruin everything for you? You know what joy is? It's a chemical signal in the brain. It's the activity of a bunch of neurotransmitters playing on your brain like a concert pianist on a set of keys. And you feel stuff. What we feel is chemical signals. Doesn't make it any less wonderful, doesn't make it any less worth seeking out joy. Go seek out joy, seriously. But cells also produce things like hormones, neurotransmitters, other chemicals that are used in certain functions in your body. So tomorrow what we are going to do is start talking about how those cellular functions occur as those cells interact with other cells and with your environment. That's it. Get out of here.